First of all, I'd like to thank the organizers for the opportunity to present some of our work at this year's OHBM. The work I'll present is focused on the heterogeneity of functional connectivity in autism. But before getting into the details about it, let's first define autism to get everyone on the same page. Autism is a neurodevelopmental disorder which is characterized by early onset difficulties in social communication and very focused, restricted interests and repetitive behaviors. People with autism also experience atypical sensory processing. And despite this core set of common symptoms among autistic people, the clinical presentation, the underlying causes, biology, and outcomes can vary greatly from person to person. Heterogeneity in autism is almost universally accepted in the field nowadays, and it poses a considerable challenge for replicating findings, discovering biomarkers, or developing targeted interventions that would be aimed at improving the outcomes for autistic people. For instance, if we look at functional connectivity literature, we can see that autism has been commonly associated with atypical functional connectivity, but the findings are largely inconsistent and sometimes contradictory, with increased and decreased connectivity being reported in the same systems. Many important studies focus on characterizing the functional connectivity in autism, but most of them are based on comparing means in a case control approach, which is perfect for well-defined groups of people, but it doesn't quite capture the individual variability among autistic people. So here we're trying to move away from comparing group means, as normative modeling allows us to model the typical functional connectivity or any other variable across age and allows us to characterize the individual deviations for whichever variable is being modeled from the norm, very much like the growth charts used by pediatricians to follow children's height and weight trajectories across age. So on one hand, autistic people converge on the core autism symptoms, which might suggest that there could be convergence of functional connectivity on at least some level of observation. But on the other hand, often inconsistent findings reported in studies of brain connectivity of autism suggest that person-specific deviations of functional connectivity may also be highly heterogeneous. So we ask the question, could different scales of analysis provide different levels of heterogeneity? To test that, we developed a way of looking at heterogeneity across multiple scales on the individual interregional connection level, the regional level, and the network level. This allows us to test different hypotheses. At the edge level, we can ask, is there a specific connection between regions that is consistently dysfunctional across subjects? Our hypothesis here is that on the individual connection level is where we see the most heterogeneity in autism that would explain away the heterogeneity in findings. Or maybe, is it the case that specific connections are consistently deviant, but they are attached to common regions? Here we hypothesize that there would be regions in which autistic people deviate positively or negatively more consistently, or that there is an overlap in deviation across autistic people, more than across controls. Our third question was, could it be the case that deviant connections aggregate in common networks? And similarly, for this question, we hypothesized that more overlap would be observed across specific networks in autistic people when compared to controls. To test this hypothesis, we use multi-scale normative modeling of resting state data from a large dataset combining data from the EU AIMS LEAP consortium and ABIDE. We first calculated the functional connectivity maps for each participant and trained a normative model on 50% of the control group to predict functional connectivity from age and sex using hierarchical Bayesian regression. We tested this model on the rest of the controls and the group of autistic people. And this is our pipeline, which was performed on 3 scales. Once we had a deviation map for each participant, we applied a threshold to identify extreme positive and negative deviations from the normative model for each person. To evaluate the degree to which deviations converged on the level of connections, we calculated the percent of people within each group that showed an extreme deviation at a given connection, and used permutation-based inference to create a null distribution, that we compared against to obtain p-values. The result didn't reach significance at any connection, as there were only less than 4% of the people that overlapped at any connection in each group. Moving forward, we wanted to look at the overlap of extreme deviations at the regional level. To do that, we counted the number of deviant connections attached to each region. And as we needed a way to binarize the regions into deviant or not deviant, and we wanted to avoid relying on a specific threshold for the deviation burden, we calculated the overlap at multiple thresholds ranging from 1 to 20 deviations attached to a region. And what this means is that each threshold we estimated the percent of people that have as many as or more deviation than the threshold. To summarize these into one measure, we calculated the area under the curve across the overlaps in each group. We then used this area under the curve to calculate the difference of regional deviation burden overlap, again using permutation by randomly shuffling the labels to create a null distribution. The result from this showed a significantly higher overlap across autistic people of both positive and negative deviations. Negative deviation overlap, which can be seen in green here, was mostly observed in somatosensory regions, anterior insula, visual cortex, the amygdala, and hippocampus. On the other hand, we observed positive deviations, which can be seen in purple here, in medial prefrontal cortex, cingulate cortex, posterior parietal regions, and subcortical regions. The difference is shown here in percent relative difference, which means, for example, that in region A, there was X% more overlap in the autism group compared to controls. No regions show significantly greater overlap in controls compared to autistic individuals. The same area under the curve procedure was used to evaluate group differences in network level overlap of functional connectivity deviations. Here we looked at percentage of extreme deviations falling within each of 7 canonical networks, additionally including subcortical structures. We then evaluated overlaps across thresholds ranging between 1 and 10% and calculated the difference between the two areas under the curve. Similarly to the region level, also here the results showed that there is more overlap across autistic people in both negative and positive deviations. And here as well, there was more negative deviation overlap in lower order sensory processing networks and their connection with attention networks as well as medial temporal lobe. Positive deviation overlap was most observed in higher order processing networks such as frontoparietal and a default mode network and the subcortical structures. No networks showed greater overlap in controls compared to autistic people for either positive or negative deviations, also for networks. So to summarize, we used a multi-scale approach to normative modeling to characterize the heterogeneity of deviant functional connectivity in autism at the level of specific connections, brain regions, and canonical functional networks. In accordance with our hypothesis, we found extreme heterogeneity at the connection level, such that no more than 4% of autistic people or controls showed an extreme deviation at the same connection. On the other hand, looking at the higher levels, overlap was considerably higher in autism at specific regions, ranging from 2 to 87%, depending on the threshold, and up to 73% when looking at networks. Collectively, these findings suggest that a core neural phenotype of autism might exist that is shared across different autistic people. It involves reduced connectivity of lower sensory motor areas and increased connectivity of higher order transmolar regions. This may reflect a breakdown of signaling within cortical processing hierarchies linking perception and associative processing. Finally, higher overlap at the region and network levels could be a plausible substrate for clinical similarities between autistic individuals, whereas heterogeneity at the connection level might explain individual differences. I'd like to end here by saying that moving forward, our work will focus on looking at how similarly overlapping individuals are similar to each other from a clinical perspective. And I'd also like to give a quick word of thanks to my supervisors, Professor Alex Pernito and Professor Jan Beiteler, and Dr. Marianne Oldehenko for their ideas, guidance, and inspiration. And I'd also like to thank our co-authors and our funders, EU AIMS and AIMS2 Trials, as well as the participants in the study. And I'd be happy to chat more about this work and answer any questions either via email, Twitter, or at my poster number 426 in Montreal.